Good morning, everyone. My name is Hasin Rehana. I'm a PhD student in Computer Sciences Department and a graduate research assistant at the Biomedical Sciences Department at University of North Dakota. Today, I will be presenting our work on cancer vaccine adjuvant name recognition from clinical trial data using large language models. These are the contents of my today's presentation. Let's start with the introduction. The objective of our study is to identify and extract adjuvant names from clinical trial data to enhance cancer vaccine research. Adjuvants are critical components that enhance the immune response to vaccines. Identifying adjuvant names in cancer vaccine clinical trial data is essential for advancing research and improving treatment outcomes. Manually curating adjuvant names from the rapidly growing biomedical literature is very challenging because the literature is growing very fast. So our goal is to leverage advanced large language models for cancer vaccine adjuvant name recognition. So let's talk about what is vaccine adjuvants. A vaccine adjuvant is a substance added to a vaccine to enhance the immune response of the vaccinated individual. It helps in producing a stronger, longer-lasting immunity against infections compared to the vaccine alone. The adjuvants are critical in developing effective vaccines, particularly for challenging pathogens where a strong and durable immune response is very necessary. The history of vaccine adjuvants dates back several decades. It started in 1926, and till date, there are a very few number of adjuvants. Here you can see a timeline of major events in the research history of vaccine adjuvants. There are a few adjuvant database and resources available out there. Among them, AdjuvaDB, VaxJo, Vaccine Adjuvant Compendium are very important ones. AdjuvaDB is a web-based comprehensive database for candidate adjuvant compendium in the clinical data. It contains compositions, functions, and other features of potential adjuvants. VaxJo is a web-based central database and analysis system that curates, stores, and analyzes vaccine adjuvants and their usage in vaccine development. Basic information about vaccine adjuvants stored in VaxJo includes adjuvant names, components, structures, appearances, storage, preparation, function, safety, and vaccines that use those adjuvants. The Vaccine Adjuvant Compendium lists points of contact for each vaccine adjuvant, enabling vaccine researchers to contact the individual intellectual property holders directly. Each vaccine record contains vaccine ontology ID, properties of that adjuvant, preclinical usage and associated publications, clinical usage and associated publications, product grade and formulation, etc. There are a few challenges of vaccine adjuvant name recognition from clinical trial data. First of all, clinical trial data often contains complex and specialized medical terminology. The context in which the adjuvants are mentioned can vary greatly. They may be referenced in different sections of the clinical trial data. We can see that in an example I'll provide later. Besides, there is often a lack of standardization in how adjuvants are named and described in the clinical reports. Adjuvant names can be ambiguous, with some names potentially referring to different substances depending on the context. And combining clinical trial data with other sources of information, for example, scientific literature databases, to improve the adjuvant name recognition can be challenging due to its differences in data formats and terminologies. Now, let's talk about large language models. Large language models are a type of deep learning algorithms that applies neural network techniques with billions and trillions of parameters on language data. The processes and understands human languages on or text using self-supervised learning techniques. Examples of some large language models are generative pre-trained transformer, which we call GPT, introduced by OpenAI, large language model Meta AI by Meta, which we call Llama. Llama has Llama 2, Llama 3, and bidirectional encoder representation of transformers, which we call BERT by Google. And there are some other large language models out there. Popular applications of large language models are text generation, translation, summarization, question answering, machine programming, machine coding, etc. In today's presentation, I will mainly focus on the GPT and Llama-based models. Our methodology involves several key steps. Here you can see two different parts. Actually, one is for without fine-tuning, the left one, and the right one is for with fine-tuning methodology. So for GPT-based model, we did not perform any kind of, fine-tuning. So we took the clinical trial data and we pre-processed them and applied GPT 3.5 and GPT 4 to predict the adjuvant names. For fine-tuned Llama-based models, we took the clinical trial data, and after data pre-processing, we divided the clinical trial data to two different parts. One is used for fine-tuning the Llama 3 models, and then the rest is used for testing the model and predict the adjuvant names. We have used Llama 3 8 billion model and Llama 3 8 billion instruct model for this task. Here you can see two examples of clinical trial data from clinicaltrial.gov website. The first one, it has the adjuvant name in the trial title already. It says that GP96 heat stroke protein peptide complex vaccine in treating patients with recurrent or progressive glioma. And it also has the same adjuvant name in some other places in the brief summary. Here you can see this is the adjuvant name. Here the adjuvant name is also repeated. And the trial also provides us with the intervention or treatment where there are the biological component and the procedure. For the other clinical trial data, you can see that the title does not mention the corresponding adjuvant name. And also in the brief summary, there is no mention of that adjuvant name, but it is mentioned in the intervention or treatment that QS21 is used for this clinical trial data. So as I said in the challenges part, that it is the first challenge is the adjuvant name can be present at different part of the clinical trial data. It is not structured like it will be present in the title, or it will be present in the brief summary, or it will be present in the interventions. Yeah, so that is one challenge. So we have used two different data sets. One is AdjuvarDB dataset, which is the main source of this data set is clinicaltrial.gov website, but it is labeled by AdjuvarDB team. And we have 97 clinical trial data in the area of cancer vaccine adjuvant from this data set. And another data set we have used is labeled by our team. It is also collected from the clinicaltrial.gov website, and it has 430 number of clinical trial data. So as I showed you in the example that the clinical trial data, we have the title, the brief description, and then the intervention names. And sometimes the summary does not provide the adjuvant name. It is in the interventions, and sometimes it is in both or in three of the parts. So we have tried two different settings. One is without intervention, like we did not provide the intervention information to the model. We only provided the title and the brief summary. And another one is with intervention. That means we provided the title, the summary, and the interventions. And this is the result for without intervention setting for GPT 3.5 and GPT 4. And we tried 0-shot setting, as well as we tried 1-shot, 2-shot, 3-shot, and 4-shot setting for GPT 3.5 and GPT 4. We have found that for GPT 3.5, 0-shot setting provide us with the highest precision of 96.82%. However, the recall and F1 score are highest for 1-shot setting. That means when we provided one example of what will be the output if this is the input. For GPT-4, we saw that for 3-shot setting, we have achieved the highest precision, recall, and F1 score. For the with intervention setting, we have found that the precision is highest for 4-shot setting with GPT 3.5. However, the recall and F1 score are highest for 1-shot setting. The recall and F1 score are highest for 1-shot setting. For GPT-4, we have seen that 0-shot, 1-shot, and 2-shot setting provide us with highest precision of 100%. However, the recall and F1 score are highest for 3-shot setting, just like the results without interventions. And the precision is not that bad. It's pretty close to 100%. Here you can see the comparative results of different large language models. We have tried GPT-4 on our manually labeled data set, and we have found the precision is 92.50, the recall is 72.07%, and the F1 score is 81.02%. And we have also tried Llama 3 with and without fine-tuning to see that how the baseline model works and how the fine-tuned model works. And we have seen that without interventions, for without intervention setting, the without fine-tuning, that means the baseline model does not perform that good. But when we provide the interventions, then the performance is somewhat good. However, the fine-tuned Llama 3 8 billion model and fine-tuned Llama 3 8 billion instruct model, both of them has good amount of precision, 85%, and the recall and F1 score are 100% and 91.89% respectively, which are the highest among all these models. And another finding of our research is when we looked into our manually curated dataset and applied GPT-4, we have found that some new adjuvants are located by GPT-4, which are not listed in the AdjuvarDB dataset yet. For example, matrix M adjuvant, CDX301, GLAAC, lipopolysaccharide, XS15, dendrite cells, ARMS, and immunomod. However, some of them are listed in other adjuvant databases, for example, VAXGeo and sometimes VAC. So we can conclude saying that GPT-4 achieved the highest precision of 99.49% when compared with the other models. The fine-tuned Llama 3 8 billion and Llama 3 8 billion instruct models exhibit highest recall of 100% and highest F1 score of 91.89%, however, showed a lower precision of 85%. And GPT-4 identified 8 new adjuvants not included in the AdjuvarDB dataset, such as matrix M1, GLAAC, and immunomod. Our study highlights the potential of large language models in advancing cancer vaccine research through accurate and efficient adjuvant name recognition. In future, we want to expand our research focus beyond cancer vaccine adjuvants to include vaccines for infectious disease. We also want to comprehensively process all the records from clinicaltrials.gov website instead of just cancer subsets And we want to utilize other open source large language models like Gemma, BERT, etc. for the same task. Thank you all for your attention. It has been a pleasure presenting our research to you today. I now invite any question you may have for our study.
Great, thank you, Hasin. Great job. Does anyone have any questions for Hasin? All right. Maybe I can ask a question, even though this is from my group. Hasin, maybe can you share your thoughts on the kind of multi-shot? Because in some model, 0-shot show the best performance, 3-shot and actually show the worst performance. So, and this is somewhat opposite to what our expectation. Can you elaborate? Can you share your thoughts on that?
So, yeah, sure. Thank you for the question. It's a very good question. So sometimes when we provide some examples, it may be very helpful, but with the increasing context information, sometimes it becomes confusing. So there is usually there remains some optimal point where it becomes optimal, and then it may deteriorate. I think for GPT-4, that optimal point is 3-shot for this specific task. Because if you see that the recall, for GPT-4, if you see that the recall is increasing, sometimes it may decrease, and then at some point it becomes the optimal. And you can see that the precision and F1 score are also optimal at this point, and then it starts to deteriorate again. So it will be something of my interest. I would like to try some more shots and see that if it improves or it just decreases from that point. But when I looked into some other literatures, I have seen that, yeah, after 3 or 4 or 5 shots, the performance improvement stops, and sometimes it deteriorates.
Okay, thank you very much.
Thank you.
All right, is there any other questions? If not, well, let's thank the speaker and move on to the next talk. Hasin, could you? Yeah, thank you.
I think that UK.
I'm sorry, what was that, Anthony?
Yuki has raised a.
Oh, okay. I'm sorry, I missed that. Hi, Yuki. Yeah, please go ahead and ask a question.
Can you hear me?
Yes, we can hear you.
Yes, I have one question. And LLM and fine-tuning show the difference in performance between precision and recall. And which do you think is better for name recognition, considering time cost for experiment?
Hasin?
Hello?
Yeah, did you hear the question?
Yeah. So among fine-tuning and without fine-tuning, I believe that fine-tuning is advantageous for us in terms of it provides us with good performance. And as we know that this is a very sensitive field, so we need to be efficient. And if we have if we have any chance that we can improve our performance, then I think we should give it a try. And for this fine tuning, as it is an open source model, the model Llama I have used, it is an open source model. So yeah, it is time consuming, but it does not cost us anything. So I believe that fine tuning is a good option.
Thank you.
Thank you.
Jungkook?
Yes, go ahead, Brian.
Yeah, I was just doing an email with some guys in my lab, and they sent over this paper here, which I could drop into the chat. But this shows, kind of so I think it's really relevant to this conversation in the last talk, because it compares fine tuning with just better prompts, using better prompts. And, this study really, I think, indicates that if you use better prompts, you can, and here's the graph that, I thought could tell us, you can, there's no fine tuning, okay? And this is GPT, so this is so very relevant to this paper, 3, 5, and then 4, so it's really great. But, intensive fine tuning with some of the open source ones and some of the other ones versus just better prompts.
Yeah.
And so I just think that this, so what I'll try to do is copy this link, right now into the chat so people can take a look. But I think it's going to be a really important one for us to look at.
Yeah, I totally agree. Thank you, Brian.
I just, and it's just one of those things, I sent a link out to my lab, about something that you guys were talking about, in an earlier talk, and they sent this back to me. So, it's just moving really fast. So I put it up there in the chat. Okay.
Got it. Appreciate it. Thank you.
Yeah.